Bismillahirrahmanirrahim. The synthesis of cholesterol is regulated through feedback inhibition, through sterol dependent mechanisms, sterol independent mechanisms, and it can also be controlled by drugs and by phosphorylation or dephosphorylation. So there is a middle-aged man on statin therapy, and he chronically consumes grapefruit juice, and this juice, the chemicals in this juice are going to have effects on the SREBP-SCAP-INSIG complex status in his cells. Then there is a man who chronically consumes diet rich in saturated fatty acids, and these saturated fats are going to affect the biochemical mechanisms, and it is going to lead towards the atherogenesis. Now the lovastatin therapy is also going to affect the status of the INSIG-SCAP complex in the cells. Now there is this man who is taking a very healthy diet rich in phytosterols, and these phytosterols are going to affect the cholesterol homeostasis through effect on the SREBP-SCAP-INSIG complex in his cells. Now this woman had, before her death, she had increased levels of phytosterols in her blood, and these were having effect on the SREBP status in her cells. Now this man is being prescribed statin therapy, and before the start of the statin therapy and after 3 months of the statin therapy, the SREBP-INSIG SCAP complex in the cells is going to be altered. It will be in a different position before the start of therapy, and in an other different position after 3 months of the therapy. Regular consumption of diet rich in saturated fats is going to affect the HMG-CoA and the LDL receptors. So coming to the cholesterol homeostasis. Cholesterol homeostasis is maintained because of its flux through the lipid metabolizing pathways, and why it is controlled? Because of the changing energy needs of the cells. Obviously, a cell which is facing starvation or fasting is not suitable for that cell to be synthesizing cholesterol. A cell which has met its energy needs and it has ATP available, reducing equivalents available, that cell is going to be involved in cholesterol synthesis. Then there is need for increased synthesis of cholesterol in rapidly dividing cells, because rapidly dividing cells are also rapidly replicating their membranes. And since cholesterol is an important component of the cell membrane, so the need for cholesterol is going to increase in these cells. And besides that, the cells maybe certain tissues need to synthesize the bile salts, still others need to synthesize the steroid hormones or the vitamin D. All these contain the steroid nucleus of the cholesterol. So in such cells there will be need for increased cholesterol synthesis. So the first mechanism for the regulation of the cholesterol synthesis is through feedback inhibition. So mevalonate, which is the 6-carbon compound formed during the stage 1, exerts feedback inhibition on the HMG-CoA reductase, and cholesterol, which is the end product, is also going to exert its inhibitory effect on the HMG-CoA reductase. Besides this, there is STEROL dependent regulation of gene expression, because nature has evolved a mechanism to control related genes through transcription factors. A single transcription factor can control multiple genes, and SREBPs or STEROL regulatory element binding proteins. These are transcription factors which regulate multiple closely related genes involved in cholesterol homeostasis. For example, gene for the HMG-CoA reductase, HMG-CoA synthase, and the gene for the LDL receptor synthesis. So SREBPs have three functional domains: an N-terminal transcription regulatory domain, 30 amino acid loop which anchors these to the membrane of the endoplasmic reticulum, and a C-terminal domain which is the cholesterol sensing domain. So you can imagine that now if you can see that this is the membrane of the endoplasmic reticulum and that is the SREBP. So in this case that would be the loop, 30 amino acid loop, which is anchoring it to the ER membrane. This may be the C for cholesterol and C terminal unit, the cholesterol sensing unit, and that may be the N-terminal or the DNA binding unit, which is the regulatory unit. So SRBPs regulate the synthesis of cholesterol. These are transcription factors. So here you can see the SREBPs embedded in the endoplasmic reticulum membrane, and their DNA binding domains and the cholesterol sensing domains are protruding into the cytosol, whereas the loop which is anchoring it to the membrane of the endoplasmic reticulum is protruding into the lumen of the endoplasmic reticulum. Now the genes for these SREBPs are present on chromosome 17 and chromosome 22, and SREBPs are closely related transcription factors formed through alternative splicing, and they are almost identical with differences in the length of their N-terminal or the DNA binding domains. So chromosome 17 gives rise to SREBP 1A and 1C involved in lipid homeostasis, whereas chromosome 22 transcribes SREBP2, which is exclusive for the cholesterol homeostasis. Besides that, these SREBPs also make arrangements for the reducing equivalents which are required for the reductive biosynthesis of the cholesterol. And reducing equivalents are going to be provided by the NADPH, and since the NADPH synthesizing pathways include the HMP shunt, and the two reactions of the HMP shunt include the G6PD or glucose 6-phosphate dehydrogenase and 6-phosphogluconolactone. So these are induced by these SREBPs, and malic enzyme also generates NADPH in the citrate shuttle. So all these malic enzyme, G6PD, 6-phosphogluconolactone formation, all these are induced by these SREBPs, which are genes for which are present on the chromosome 17 and 22. When the intracellular cholesterol is low, there is proteolytic activation of the SREBP, and the SREBP moves from the endoplasmic reticulum into the Golgi apparatus, and in the Golgi it is cleaved. First the cholesterol sensing domain bound to the SCAP is cleaved, so it is relieved of the SCAP, and next the N-terminal domain or the DNA binding domain is cleaved, and this domain then moves from the Golgi to the nucleus. After translocation into the nucleus, it induces the gene expression of the HMG-CoA reductase or the LDL receptors. So here you can see that, for example, this is the HMG-CoA reductase gene present in the nucleus, and green colored transcription factor is the SREBP2, which is concerned with the cholesterol homeostasis, and in the red color you can see there is SCAP or the SREBP cleavage activating protein. So when it is present in the ER membrane, it cannot work, its N-terminal domain cannot translocate to the nucleus. So now there is a scenario when there is low cholesterol. When there is low cholesterol, the SCAP escorts the SREBP to the Golgi where it is cleaved, and its N-terminal domain translocates. After cleavage, it translocates to the nucleus and leads to the transcription of the HMG-CoA reductase gene. So here you can see that SCAP is escorting the SREBP to the Golgi for cleavage. The other scenario may be high cholesterol. In case of high cholesterol, the SREBP-SCAP complex along with the INSIG protein is retained in the membrane of the endoplasmic reticulum. It does not translocate to the Golgi, and it cannot undergo cleavage, and it cannot induce the synthesis of the genes involved in cholesterol synthesis. So when the SCAP-INSIG and the precursor SREBPs are bound, it is the inactive complex, and when it is inactive, when there is increased level of cholesterol. So here you can see that INSIG also has the cholesterol sensing ability, plus here you can see the SCAP site for the sensing and binding of the cholesterol. Now when there is high intracellular cholesterol shown by these yellow colored molecules, the INSIG and SCAP are going to collaborate. They feel threatened by these cholesterol molecules, and these are going to lock the SREBP in the ER membrane. These are going to make this ER membrane a subjail for the SREBP. So for the sake of retaining this thing in the memory, you can imagine that these cholesterol molecules are the people. So these are the people, and the SCAP and INSIG are the lawmakers, and they feel threatened by these people, so they lock the SREBP in the ER membrane. They make it a subjail for the SREBP. Now this complex is inactive. Why it is inactive? Because they all are sitting and doing nothing. So here you can see that they all are sitting like lethargians. SCAP, INSIG and the SREBP, no one is leaving its place, and this is the inactive complex. The other scenario may be low cholesterol. So cholesterol moves away, means that this crowd of people moves away. Now the SCAP and INSIG are relaxed. They enter collaboration and they relieve this SREBP from the subjail. Now the SREBP can move from the ER membrane to the Golgi. because the cholesterol is low. So here you can see that the INSIG and SCAP have ended their collaboration and the SREBP moves to the golgi. Here it is going to undergo cleavage. The C-terminal domain is going to be cleaved by the S1P or site 1 protease, and now SCAP leaves it on its own. Now SCAP is not holding its hand. Next, the N-terminal domain needs to be cleaved, and for this S2 protease S2P comes into action and it cleaves the N-terminal domain. Now this N-terminal domain moves to the nucleus. After translocation to the nucleus, it induces the transcription of the gene for the synthesis of the HMG-CoA reductase so that cholesterol can be synthesized. Not only that, if for example HMG-CoA is synthesized, it escapes the inhibition or it has already started synthesizing the cholesterol, then what happens? Then it can be degraded, and the degradation is STEROL accelerated enzyme degradation. Here you can see this is the INSIG complex, and that is the rust colored HMG-CoA, and this oval is the catalytic unit of the HMG-CoA protruding into the cytosol. So when there is increased cholesterol, the INSIG is going to sense that there is increased cholesterol, and it is going to cause ubiquitination of the HMG-CoA represented by these blue colored molecules. At two sites of HMG-CoA, it is going to cause the attachment of these ubiquitin. So now it has been tagged for destruction. It has been granted a visa to the hereafter. So what will happen? It will move to the proteasosome for degradation. It will be degraded to its constituent amino acids, and now this HMG-CoA is like a trash. Okay. Sterol independent mechanisms also control the cholesterol homeostasis through phosphorylation and dephosphorylation, and insulin and glucagon play a role in it. Besides insulin and glucagon, thyroid hormone and glucocorticoids also affect the cholesterol synthesis. Thyroid hormone increases the activity of HMG-CoA reductase, whereas glucocorticoids decrease the activity of the HMG-CoA reductase. So here you can see this is the HMG-CoA reductase. In presence of glucagon and AMP activated protein kinase, it is phosphorylated. So the phosphorylated HMG-CoA reductase is inactive. And how can you remember that it is inactive? Because it is carrying the weight of the phosphate. Inactive HMG-CoA reductase can be activated by insulin in the presence of protein phosphatase by dephosphorylation. So the phosphorylated HMG-CoA is carrying the load of phosphate, and therefore it is inactive. Phosphate is too heavy for it. Whereas the dephosphorylated HMG-CoA reductase is agile and active, and it can play its part in cholesterol synthesis. So here you can see that insulin binding to the tyrosine kinase receptors leads to the phosphorylation of the SREBP1C, leading to the synthesis of the enzymes involved in cholesterol and lipid metabolism. So now coming to the MCQs. SCAP-INSIG complex retains SREBP in ER membrane when the cholesterol level is high. SCAP-INSIG complex dissociates from and allows SREBP to move to golgi when the cholesterol level is low. Now revisiting the waiting room. Effect of the grapefruit juice consumption, chronic consumption of grapefruit juice on the statin therapy. The grapefruit fruit contains furanocoumarins. Furanocoumarins inhibit the cytochrome P450, CYP3A4, and this enzyme was P450 is involved in xenobiotic metabolism, and it helps in the excretion of the statins. Now when this is inhibited, statins will build up in the blood, so there will be increased inhibition of the cholesterol synthesis, and this will lead to low intracellular cholesterol. So when the intracellular cholesterol is low, the SCAP-INSIG complex will end their collaboration for the retention of the SREBP into the endoplasmic reticulum membrane, and it will become active. The SREBP complex will translocate to the golgi, where it will be cleaved, and the N-terminal unit will move to the nucleus for transcription of LDL receptors, so that they can catch the cholesterol from the blood and bring it inside the cell. Now for someone who is on statin therapy, what will happen to the SCAP-INSIG complex? Someone with statin therapy is going to have low intracellular cholesterol. So when the cholesterol is low, the SCAP-INSIG complex will become active. They will end their collaboration. SCAP will escort the SREBP to the golgi, and here it will be cleaved, and N-terminal unit will translocate to the nucleus for the synthesis of the LDL receptors. So this is the diagrammatic representation that acetyl-CoAs from the glucose or from the beta oxidation of the long chain fatty acids are used for the synthesis of cholesterol. So the HMG-CoA reductase leads to the formation of mevalonate, which ends up after many steps in the synthesis of cholesterol. And this cholesterol is going to contribute to the cholesterol pool inside the cell. When statins inhibit this HMG-CoA reductase, the pathway for the cholesterol synthesis stops, leading to low cholesterol inside the cell. This leads to the activation of the SREBP and its cleavage and the translocation of the N-terminal unit to the nucleus. And transcription of the gene and LDL receptor synthesis. So LDL receptors are going to catch they are going to catch either the chylomicron remnants or the LDL particles, and these will be endocytosed. Here you can see that clathrin coated pit is helping in the endocytosis. Clathrin is recycled. The LDL receptors are also recycled, whereas this LDL particle is digested. Amino acids, fatty acids, and the cholesterol is released into the cell. So this is what is taking place inside the cells of someone on statin therapy. Now if someone is taking lots of phytosterols, what will happen to the SCAP-INSIG complex? The phytosterols help in the lowering of the intracellular cholesterol. How? That from the intestine, when we take in fatty diet, emulsion droplets are formed, which are finally converted to micelles, and the micelles dock on the brush border, and their contents are translocated into the enterocyte, and the cholesterol also comes in the enterocyte through NPC1 protein. Now once it is inside this enterocyte, the phytosterols bind to the cholesterol, and along with the cholesterol, these are thrown back into the intestinal lumen through the ABC cassette proteins ABC, G5 and G8. So this leads to the lowering of the cholesterol level in the blood. So someone taking lots of phytosterol is going to have low intracellular cholesterol. So with low intracellular cholesterol, SREBP-SCAP-INSIG complex is going to be active. SREBP complex will move to the Golgi, it will be cleaved, the N-terminal unit will move to the nucleus, cause the transcription of the LDL receptor, so that they can catch the LDL particles moving in the circulation and bring them inside the cell for the replenishment of the cholesterol pool inside the cell. So here you can see these are effluxed, thrown back into the intestinal lumen. Okay. Now for someone who regularly consumes diet rich in saturated fats, small LDL particles are small VLDL particles with more cholesterol are going to be formed, and there will be their slow utilization, and this is going to contribute towards atherogenesis. Now this female who died of phytosterolemia, during her lifetime she was having increased levels of cholesterol in her cells, and therefore her SCAP-INSIG SREBP complex was inactive. SCAP and INSIG were collaborating to keep the SREBP locked in the ER membrane. So to summarize, three main processes regulate the cholesterol homeostasis. First is the endocytosis of LDL particles by the LDL receptors. Second is the de novo synthesis of the cholesterol by liver enzymes, and third is the stimulation of the LDL receptor gene expression.